Imagine a bustling city, its intricate network of roads representing our DNA. This is a micronucleus, a tiny aberrant structure within our cells, separate from the main nucleus. These micronuclei are telltale signs of genomic instability, a hallmark of cancer. Recognizing them is crucial. They serve as red flags, alerting us to potential cancer development. Understanding their biology can unlock new strategies to combat this disease. The formation of a micronucleus is rarely a deliberate act. It is often a consequence of errors during cell division, a process known as mitosis. Picture a delicate dance, chromosomes meticulously dividing to create two identical daughter cells. Occasionally, this intricate choreography falters. Chromosomes lag behind, failing to integrate properly into the newly forming nuclei. These stranded chromosomes become enveloped in their own membrane, forming a micronucleus, a separate entity within the cell. Several factors can disrupt this delicate dance, increasing the likelihood of micronuclei formation. Exposure to radiation, certain chemicals, and even inherent genetic predispositions can all contribute to these mitotic mishaps. POS3S P62 is a protein that has its own life, Santaguida said. It works in the cells by recognizing proteins or organelles that have to be degraded, targeting them for a process called autophagy. Santaguida's team found that ROS from mitochondria also alter P62, ramping up its activity to send proteins down the autophagy pathway. This overreactive P62 degrades the repair enzyme SCAR-T3, further dooming the micronuclear membrane to collapse. To connect P62 with cancer prognosis, the team analyzed data from the tumors of 875 gastric cancer patients and found that higher levels of P62 were correlated with shortened survival. The higher the levels of P62, the higher the likelihood to get a worse prognosis, Santaguida said. It's a prognostic marker that can be employed in the clinic tomorrow. Once formed, a micronucleus faces a critical juncture, persistence, or degradation. Some micronuclei are quickly eliminated by the cell's quality control. However, some evade these defenses. They persist, undergoing flawed DNA replication. This can drive the cell towards malignancy. Genomic instability is a state of constant flux within a cell's DNA. It's akin to a city's infrastructure constantly undergoing haphazard repairs, leading to a chaotic and unreliable network. This instability is a hallmark of cancer, enabling cells to acquire mutations that fuel their uncontrolled growth and spread. The presence of elevated micronuclei levels serves as a red flag, signaling heightened genomic instability. These miniature worlds, harboring damaged and missegregated chromosomes, contribute to the overall disarray within the cell's genetic blueprint. This link between micronuclei and genomic instability is well established. Studies have shown a strong correlation between elevated micronuclei frequencies and an increased risk of developing various cancers. This underscores their significance as both biomarkers and drivers of tumorigenesis. Fueling the fire: how micronuclei drive cancer. Micronuclei are not merely passive bystanders in the development of cancer. They actively contribute to the disease's progression through various mechanisms, acting as catalysts that accelerate tumor growth and spread. Chromothripsis, shattering the blueprint. Imagine a city struck by a devastating earthquake, its infrastructure shattered into countless fragments. This catastrophic event mirrors the phenomenon of chromothripsis, a process triggered by micronuclei. During chromothripsis, chromosomes within micronuclei can undergo massive fragmentation and chaotic reassembly. This results in a scrambled genome with genes shuffled and rearranged in a haphazard manner. This genomic chaos can have profound consequences. It can activate cancer-promoting genes or silence tumor suppressor genes, effectively removing the brakes on uncontrolled cell growth. Fanning the flames, micronuclei, and inflammation. Inflammation is a double-edged sword. While crucial for fighting off infections, chronic inflammation can create a fertile ground for cancer development. Micronuclei, surprisingly, can fuel this inflammatory fire. When micronuclei rupture, they release their fragmented DNA into the cell's cytoplasm. This triggers an immune response, as the cell recognizes this DNA as foreign and potentially dangerous. This chronic activation of the immune system, driven by micronuclei, contributes to a pro-inflammatory environment within the tumor microenvironment. This, in turn, promotes tumor growth, angiogenesis, and even resistance to therapy. Cheating death escaping apoptosis. Apoptosis, or programmed cell death, is a crucial mechanism for eliminating damaged or potentially harmful cells. It ensures that cells with irreparable DNA damage do not persist and contribute to tumor development. However, cancer cells often evolve ways to evade apoptosis, allowing them to survive and proliferate unchecked. Micronuclei, surprisingly, can play a role in this escape act. The presence of micronuclei can interfere with the normal signaling pathways that trigger apoptosis. This allows cancer cells to bypass this critical checkpoint, even in the face of significant genomic damage, further fueling tumor development. Aneuploidy, an unbalanced equation. Aneuploidy, an abnormal number of chromosomes within a cell, is a hallmark of cancer. This imbalance disrupts the delicate equilibrium of gene expression, driving tumorigenesis. Micronuclei contribute to aneuploidy by generating daughter cells with an uneven distribution of chromosomes. This occurs because the genetic material within micronuclei is often not equally segregated during cell division. This aneuploidy further fuels genomic instability, providing a constant source of genetic variation upon which natural selection can act. This drives tumor evolution, leading to more aggressive and therapy-resistant cancer cells. Turning the tide targeting micronuclei in cancer therapy. The intricate link between micronuclei and cancer progression presents a unique opportunity. By understanding and exploiting the biology of these miniature worlds, we can develop novel therapeutic strategies to combat this devastating disease. Sentinels of instability, micronuclei serve as valuable biomarkers. The presence of elevated micronuclei levels provides insights into an individual's risk of developing cancer and their response to therapy. By monitoring micronuclei frequencies, clinicians can identify individuals at higher risk and tailor screening and prevention strategies accordingly. Similarly, changes in micronuclei levels during treatment can provide valuable information about a tumor's response to therapy. This personalized approach, guided by micronuclei analysis, allows for more effective cancer management and improved patient outcomes. Disarming the alarm, the cGAS sting pathway. As previously discussed, ruptured micronuclei activate the cGAS sting pathway, triggering an inflammatory response that can promote tumor growth. This pathway presents an attractive therapeutic target. By developing drugs that inhibit the cGAS sting pathway, we can potentially dampen the inflammatory response triggered by micronuclei. This could help limit tumor growth and improve the efficacy of other cancer therapies. This approach is still in its early stages, but it holds promising potential for developing novel cancer treatments. Embracing destruction, enhancing micronuclei-induced death. While some cancer cells evade apoptosis despite the presence of micronuclei, others are more susceptible to cell death triggered by these genomic anomalies. This vulnerability presents another therapeutic avenue. Researchers are exploring ways to enhance micronuclei-induced cell death. By developing drugs that exploit this vulnerability, we can selectively target and eliminate cancer cells harboring these genomic time bombs. This targeted approach could minimize damage to healthy cells while maximizing the impact on cancer cells, leading to more effective and less toxic therapies. Exploiting the chaos chromothripsis as a weapon. While chromothripsis, triggered by micronuclei, often benefits cancer cells, it can also be their Achilles heel. The extensive genomic rearrangements caused by this phenomenon can create vulnerabilities that we can exploit. Researchers are investigating ways to identify and target these vulnerabilities, developing therapies that specifically target cancer cells with chromothripsis-induced genomic alterations. This approach holds promise for developing personalized cancer treatments tailored to the unique genomic landscape of each patient's tumor. A personalized approach, tailoring treatment to micronuclei. The presence and behavior of micronuclei can vary significantly between different cancer types and even within individual tumors. This highlights the need for a personalized approach to cancer treatment, guided by an understanding of micronuclei biology. By analyzing the specific characteristics of micronuclei within a patient's tumor, clinicians can tailor treatment strategies accordingly. This might involve selecting therapies that exploit micronuclei-induced vulnerabilities or monitoring micronuclei levels to assess treatment response. This personalized approach, driven by an understanding of micronuclei, has the potential to revolutionize cancer treatment and improve patient outcomes. A new dawn, micronuclei as beacons of hope. Micronuclei have opened new avenues in cancer biology. Once overlooked, they are now recognized as powerful drivers of tumorigenesis and valuable biomarkers. Insights from studying micronuclei provide hope. By harnessing this knowledge, we can strive towards a future where cancer is manageable. Continued research and collaboration can turn the tide against cancer. In 2022, on the alpine shores of Lake Maggiore in northern Italy, longtime friends Samuel Bacum, Maryland, Ph.D., and Stefano Santaguida, Ph.D., were catching up. Pandemic restrictions had prevented the two from seeing each other for years, but at a small conference in the lakeside town of Stresa, they seized the opportunity to share what they'd been working on recently: the collapse of micronuclei in cancer cells. "We were so happy and excited to be together in real life," Santaguida, a molecular biologist at the European Institute of Oncology and the University of Milan, told Fierce Biotech in an interview. "We were working on two separate aspects at the very beginning, without knowing of each other's work. This serendipitous collaboration brought fresh perspectives and combined expertise, accelerating the understanding of micronuclei in cancer. It serves as a testament to the power of human connection and shared passion in overcoming one of the most challenging diseases of our time." As we continue to explore the mysteries of micronuclei, such collaborations will be the cornerstone of breakthroughs, driving us ever closer to a world where cancer is no longer a formidable foe, but a manageable condition. As the understanding of micronuclei continues to deepen, the future of cancer research looks more promising than ever. Teaming up to tackle the problem has now resulted in two landmark papers published on August 29 in the journal Science. These studies shed light on the intricate mechanisms behind micronuclei rupture and collapse in cancer cells, a pivotal process that accelerates cancer progression. By entangling with mitochondria, micronuclei become vulnerable to reactive oxygen species, which alter the behavior of crucial repair proteins and overactivate protein-destroying pathways. A defining trait of cancer cells is aneuploidy, where cells possess an abnormal number of chromosomes. This anomaly occurs during cell division, when some chromosomes fail to integrate into the nucleus of the new daughter cell. These stranded chromosomes form flimsy nuclear membranes around themselves, resulting in micronuclei that are smaller and less functional than the primary nuclear envelope. Consequently, these micronuclei can rupture, exposing the chromosomes to cellular elements. That rupture process underlies a lot of the means by which micronuclei seem to drive cancer progression, explains Samuel Bacum, a cell biologist at Memorial Sloan Kettering Cancer Center in New York City. With each discovery, researchers are not only unraveling the complexities of cancer, but also paving the way for innovative therapies. The collaboration between experts like Bacum and Santaguida exemplifies how shared knowledge and passion can lead to groundbreaking advancements. As we continue to explore the mysteries of micronuclei, the collective efforts of the scientific community will be instrumental in transforming cancer from a formidable foe into a manageable condition. The journey is far from over, but with every step forward, we move closer to a future where cancer's grip on humanity is significantly weakened. The promise of new treatments and a deeper understanding of the disease offers hope to millions around the world. As the landscape of cancer research evolves, micronuclei have emerged as a beacon of personalized medicine, offering a new frontier in the battle against cancer. Enzymes in the cytoplasm break chunks off the free-floating DNA, leading to mutations that can spur new cancer traits or alter how genes are expressed. This dynamic process not only accelerates tumor evolution, but also presents unique biomarkers for tailored therapies. Melody DeBona, Ph.D., a cancer biologist working in Bacum's group, was observing cervical cancer cells under the microscope when she noticed that micronuclei tended to get tangled up with mitochondria. "My hypothesis when I started to look at cells was that mitochondria were actually entering the micronucleus," DeBona told Fierce in a joint interview with Santaguida. Investigating further, she instead found that the chance of rupture greatly increased, as the micronucleus was exposed to more and more toxic forms of oxygen-containing molecules leaking from the mitochondria, called reactive oxygen species, ROS. Through a series of experiments, she pieced together how the membrane unraveling unfolds. The implications of these findings are profound. The cell can confuse the loose chromosome for a virus, sparking inflammation that can lead the tumor to evolve immune resistance. Understanding these mechanisms opens the door to novel therapeutic strategies that target the unique vulnerabilities of cancer cells. Imagine a world where cancer treatment is no longer a one-size-fits-all approach, but is instead tailored to the specific genetic and cellular anomalies of each patient's tumor. Micronuclei research is paving the way for this reality, offering hope and precision in the fight against cancer. As we stand on the cusp of this new era, the collaboration and dedication of scientists around the world are driving us closer to turning the tide against cancer. The journey is challenging, but with every discovery, we move a step closer to a future where personalized medicine transforms the lives of millions. Stay tuned as we continue to explore the groundbreaking advancements in micronuclei research and their potential to revolutionize cancer therapy. Tumors tend to be low on oxygen, which the mitochondria normally need to function. When oxygen is low, nitric oxide binds in its place instead, producing reactive oxygen species, or ROS, as a byproduct. ROS are unstable molecules that can steal electrons from other molecules, disrupting their form and function. Debona discovered that ROS take electrons away from amino acids called cysteines in the protein CHMP7, altering its shape so that it abnormally binds to another protein called LEMD2, situated within the micronuclear membrane. This binding causes a cascade of cellular chaos. What's more, ROS damage also prevents CHMP7 from being expelled from the cell. LEMD2 is already in the membrane, Bakum explains. Now imagine a protein that's aggregating inside, right underneath, and pulling it away. The force of CHMP7 tugging at LEMD2 causes the micronucleus's membrane to collapse and rupture. Debona's interest in CHMP7 began because it is typically a crucial component of a nuclear membrane repair enzyme known as SCAR-T3. However, this enzyme is hindered from repairing the rupturing micronucleus due to the actions of another protein called P62. This revelation underscores the intricate interplay between cellular components and the devastating chain reactions that can drive cancer progression. Understanding these molecular dynamics provides invaluable insights for developing targeted therapies aimed at mitigating the chaos within cancer cells. Stay with us as we delve deeper into the groundbreaking advancements in micronuclei research and their potential to revolutionize cancer therapy, one discovery at a time. In addition to the prognostic power of P62, the collapse mechanism presents several other potential targets for new drugs. Therapies could target cysteines in CHMP7 and push them away from a pathological path, Bakum said. And numerous other molecules play bit parts in the process too, like kinases in the nuclear factor Kb pathway. Targeting them could reduce the odds of micronuclei rupturing and causing cancer progression. Chromosomes in cancer cells are starting to attract more attention from biotechs. Bakum is a co-founder and scientific advisor of Velastra Therapeutics, a biotech focused on chromosomal instability, and also serves on the scientific advisory board of precision oncology company Meliora Therapeutics. We can't suppress chromosome instability in cancer because the cat's out of the bag, but can we at least kneecap it? Bakum said. Can we intervene in a way that prevents all the bad consequences and sequelae of micronuclear rupture? As we continue to explore these emerging therapeutic strategies, the future of cancer treatment looks increasingly promising. Research and innovation in the realm of micronuclei could pave the way for revolutionary approaches to managing and potentially overcoming cancer. Stay tuned as we delve into the collaborative efforts and cutting-edge technologies driving these advancements. In conclusion, micronuclei play a crucial role in cancer biology. From their formation due to cellular errors to their impact on cancer progression and therapy, these tiny structures are pivotal. Key points include 1. Micronuclei formation and persistence. 1. Their role in driving cancer and inflammation. 1. Potential as biomarkers and therapeutic targets. Understanding micronuclei opens new frontiers in personalized medicine and cancer treatment. Stay informed and hopeful as research continues to unveil their mysteries.